Hello everyone, welcome back to my channel IGCSE Biology Help. In today's video, I'm going to be solving the October-November 2022 paper 4 variant 1. This is the extended paper. Make sure to subscribe to my channel and like my videos to receive more videos like this one. Let's start solving. Question number 1a, figure 1.1 is a side view of a human skull indicating the 4 types of teeth and the jaws. State the function of the human teeth. Of course, it is mechanical digestion. You can also say physical digestion if you want. Question double I, state the name of the visible outer layer of the teeth. It is the enamel. Triple I, explain the process of tooth decay in humans. Of course, it starts off with the human or the person eating sugary sweets or foods that contain sugar. This sugar is going to accumulate on the teeth, and then respiration is going to occur by anaerobic bacteria. These anaerobic bacteria will produce lactic acid. This acid, of course, will dissolve or break down the enamel. This will cause tooth decay, and this will cause the dentin and other layers of the teeth to be exposed. B. Mammals can be classified according to the position and shape of their teeth. Figure 1.2 shows the skulls of 7 mammals. I, use the key to identify each species shown in figure 1.2. Write the letter of each species A to G in the correct box in the key. Now this is the question that is called dichotomous key. This question, actually, you have to follow each of the, for example here it's like go to 2, go to 3. You have to follow what is written over here. 1a says 2 or more different types of teeth. Go to 2. Now we're going to be going to 2, which is this one. 2a says have a wide gap between front and back teeth in both jaws. So we have to look first at the species over here that have two different types of teeth. I can see that this one has 2 different types of teeth, as well as this one, and this one, and this one as well. So we have A, C, D, and G. So have a wide gap between front and back teeth in both jaws. Go to 4. We can see that this one has a wide gap, as well as this one. So let's see. Over here it says go to 4. So right now I'm in 4. No incisors in upper jaw. This is going to be G. So this one has no incisors on the upper jaw over here. There's no incisors. So this is the first one. We answered it. Now let's see the other one. It says incisors in both upper and lower jaw. Got to 5. So I can see that this one over here, A, has incisors on both the upper and lower jaw. A, so it says go to 5. I'm in 5 now. Incisors on lower jaw longer than incisors on upper jaw. Over here this is actually incorrect because they're both equal sizes. So A over here is not matching the description. Incisors on upper and lower jaw are similar in size. This is A, so this is actually matching the description. They are similar in size, but I can't say that they are the same size, but they are similar. As we can see, they're not different sizes. So A is the correct description for this one. So let's go back over here to this one, fewer than 2 different types of teeth. Over here we can see that this is actually E, and F, it has no teeth at all, and B as well. So let's see over here. It says go to 3. So this is 3. 3A, all teeth of similar shape. This is actually over here E. We can see that all the teeth are jagged like this. There is no different types of teeth at all. So this is E. We are done with it. No teeth on either jaw. This is actually F. So this is also answered. So let's continue over here. Let's see the other option, the 2 or more different types of teeth. Let's continue. We finished this one too. Let's see the other option in 2. B says have no wide gap between front and back teeth in both jaws. So let's see over here which ones do not have wide gap. This is actually C, this one, and also D, and that's it. It's only C and D. So let's see. It says go to 6. Incisors on lower jaw project forwards. So this is actually matching the description of C. I can see over here that there's this one incisor that is pointing forwards like this. So this is going to be C. And then B says incisors on lower jaw do not project forwards. Of course, this is going to be D. They do not have anything that is pointed like C. So this is going to be D. So we answered this one. And let's see, there is only one that remains unanswered, and this is actually 5A. Incisors on lower jaw longer than incisors on upper jaw. This is going to be B, of course, because we can see that this one is so much longer than the upper one. Double I, killer whales, a species, are mammals. State 2 internal features you would expect to find in a killer whale that you would not find in a fish. This is going to be killer whales are mammals, and fish actually do not have double circulation. We all know that fish has a single circulation, and they don't have a four-chambered heart or lungs. They have gills instead. These act as lungs for the fish, but they are not lungs. And of course, they don't have a diaphragm because this is something found only in mammals. Triple I, state the name of the group of animals that includes mammals and fish. Of course, this is the vertebrates. 2. Digestive enzymes catalyze the breakdown of large insoluble molecules. A says, explain why it is important that large insoluble molecules are broken down by chemical digestion. So, of course, this is for the molecules to become soluble for absorption, which means that if you just have a big chunk of food that you just ate and you just swallow it down, it will not be small enough for your stomach to absorb or the blood to absorb, and it's going to be too big for the minerals to be extracted from it. So it has to be soluble, and also it should be small enough for reactions such as diffusion or active transport. Double I, state the name of the substance that is the solvent for most molecules that have been digested by enzymes. This is actually water. B, the activity of two protease enzymes A and B was measured at different pHs. Both enzymes are found in the human alimentary canal. The results are shown in figure 2.1. Describe and explain the roles of the two protease enzymes A and B in the alimentary canal. Use the information in figure 2.1 to support your answer. So this question is going to be description and explanation. So you must write down points for description and explanation. You should not leave out anything. So number 1 is actually an explanation. It says that both of the enzymes turn protein into amino acids because they are protease enzymes. Number 2 says refer to specificity. This means that, for example, you're going to say that the enzyme active sites binds to complementary substrate shape. And we can also say that 3, both enzymes are active between pH 3 and 5. 3 is actually a description because we are saying something that is on the graph. You can see over here that yes, both of them are active between pH 3 and 5. And we can also say, for example, that enzyme A is active in pH 4 at pH 4, and we can also say, for example, that enzyme B is active, for example, in pH 5. This is a description. We can say, I can see also that enzyme A is quite active in acidic pH. This means that this is a stomach enzyme, for example, like pepsin. So we can also add this point. We can also, I can see, I wrote this point down that A is a pepsin because it is active in acidic pHs. You can also say this point. You can also say that enzyme shows no activity or is denatured from pH 5. This is talking about the pepsin enzyme, enzyme A. It does not show any type of activity when it is when the pH is increasing after pH 5. This is because this enzyme works its best in acidic pH. It is an acidic enzyme. Points are written down as separate points. If you write a sentence, they don't mark you for a sentence, they mark the keywords. They look for the keywords in your answer. So the keywords are actually the things that help you in your answer. You can also say that B is a trypsin because it is actually active in alkaline pH. It is the opposite of enzyme A. And you can also say that it shows no activity. It is denatured from pH 3 because it is an alkaline enzyme. C says maltase is a digestive enzyme that acts in the small intestine. State the exact location of maltase in the small intestine. Of course, this is the epithelium. 3a, figure 3.1 shows some apparatus that was used to investigate water loss from a leafy shoot. I say state the name of the process by which leafy shoots lose water. Of course, this is the transpiration. Double I, before the leafy shoot is inserted into the jar, shown in figure 3.1, it must be recut underwater. State why the end of the leafy shoot was cut underwater. Of course, this is a question that comes in many exams. The answer is always to prevent air bubbles or ensure continuous column of water. Triple I, state the purpose of the oil layer on top of the water. This is to prevent evaporation or condensation. IV, using the information in figure 3.1, describe one method that can be used to determine how much water is lost from the leafy shoot. This can be done by two methods. It is by measuring the decrease in the volume of the water in a burette over a period of time. This can be done, for example, if you have a burette in the start of the experiment, you can record the amount of water or measure the amount of water and write it down. After the experiment, you can, for example, check the water or you can measure the water after the experiment in the burette and calculate the difference to know how much water is lost. You can also do another method that is a bit simpler. It is to record the decrease in mass over time. This can be done, for example, by measuring the mass of the leaf before the experiment and after the experiment, and you're going to minus both of these numbers to get the difference. B, the apparatus shown in figure 3.1 was used to investigate the effect of temperature on the rate of water loss in a species of a plant. The results are shown in figure 3.2. Using the information in figure 3.2, calculate how much water would be lost from 1 meter squared of leaves in 12 hours if the plants were kept at 35 degrees, include the unit. So over here we can see that at temperature 35, we can see that there is 0.28 grams per meter cubed per second. So over here, this is in 1 second. If you want to calculate in 12 hours, you're going to multiply this number 0.28 by 62 times. This is because you're going to find out the number of seconds. 60 times 60, this is the number of seconds in 1 hour, times 12, because there's 12 hours. This is going to give you 12,096 grams. Double I, using the information in figure 3.2, describe and explain the effect of increasing temperature on the rate of water loss in the species of plant. So this question wants us to describe what's in the graph and also explain how this can have occurred. So first of all, it says as the temperature increases, the rate of water loss increases. We can see over here the graph that as the temperature increases, the graph goes up and it becomes steeper. So this is a proof of the point. Another one is I can see that after 35 over here, we can see that it became much steeper, or actually at 37 I can see that it's after 37 the rate of water loss has become much steeper and it increased very much. We can say any data code, which means you can say, for example, at 35 degrees over here, The water loss rate was 0.28 per second. You can say that. You must, of course, include the units to get a mark. 4 says water vapor lost through stomata between guard cells. So this is the explanation of how there is a water loss in the leaf. This is basically an explanation of transpiration. 5 evaporation from mesophyll into airspaces. 6 diffusion down a water potential gradient. And lastly, increasing temperature increases kinetic energy. C. The apparatus shown in figure 3.1 can also be used to investigate the effects of changing humidity on water loss in plants. Suggest why the mass of water in the apparatus does not change when the leafy shoot is kept at 100 relative humidity. So if the humidity is at 100, there is going to be no diffusion, there is no water potential gradient, so this will not lead to any change in the mass of the water of the leaf. Double I, even at extremely low relative humidities, the leafy shoot did not wilt. Explain why the leafy shoot shown in figure 3.1 did not wilt. So this is actually figure 3.1 over here. This is the leafy shoot they are talking about. So we can say that there is a continuous supply of water that is provided. As we all know that wilting occurs because of a loss of the water, or the cell becomes the plant cells becomes plasmolized. This is because there is absence of water in the leaf. This can occur when there, for example, if you have a plant at home and you don't water it, so it will wilt and dry. So we all saw this before. This is the reason of wilting. So if there is water provided, of course there will not be any wilting. Triple I, the investigation on the effect of temperature was done at a relative humidity of 20%. The investigation was repeated at a relative humidity of 80%, and all other conditions were kept the same. Predict how the water loss will differ from the trend shown in figure 3.1. Sketch your prediction. So over here I have actually drawn this down. This is the one that I have drawn. It is this one below the original graph. This is because we know that the humidity, as it increases, there is going to be less water loss. There is less changes in the water mass of the leaf. So at higher humidity, there is lower transpiration. 4 chromosomes are made of DNA. Describe the structure of a DNA molecule. So this is, of course, we can say that there is 2 strands formed into double helix with 4 different types of bases on them, A, C, T, and G. The A binds with the T and the C binds with the G. There is cross links between bases. This is a question that is memorized. BI, outline how antibiotic resistance develops in a population of bacteria. First of all, there is variation on the bacteria, and this will cause mutation so that the bacteria can adapt to the environment. Mutations give bacteria an advantage. Non-resistant bacteria are killed. So the bacteria basically mutate so that they can survive. And of course, this mutation will spread to other generations of bacteria, because only the resistant bacteria actually reproduce. So the mutation will pass on to future generations of the bacteria. So then the competition with non-resistant bacteria occurs. So then of course the resistant bacteria will win, because they will reproduce rapidly, because they can survive in the environmental pressure that they're in. Double I, scientists use differences in antibiotic resistant genes to distinguish between different strains of the bacterium and MRSA. Suggest why scientists use differences in base sequence to classify the strains of MRSA rather than using other methods. This is because it is actually more accurate, and you do not need to have a large quantity of the bacteria. So this is something good if, for example, the bacteria is extinct, or it is there is no large amount of this bacteria available, and there is no need to culture the bacteria. So again, this leads us to the last point. Triple I explain why scientists are concerned that some strains of bacteria, such as SRS, have become resistant to antibiotics. Of course, this is a problem because it will cause a lot of bacterial diseases to not be cured, and it will be very hard to cure these diseases for someone who is having an antibiotic resistance. So, also, this will cause resistant bacteria to not be killed by antibiotics. CI describe how the use of antibiotics can be managed to reduce the development of resistant strains of bacteria. Of course, the first thing and the most important thing is to use antibiotics only when essential. This means you do not use antibiotics if, for example, viral infection or you have anything other than a bacterial infection. Also another way is to ensure people that they follow the instructions and complete the course of antibiotics. This means, for example, if a doctor prescribes an antibiotic for example, a week, then the patient has to follow all the rules and they should finish the antibiotic even if they feel better. Suggest why MRSA is unlikely to be transmitted from a mother to her unborn fetus. This is because the placenta forms a barrier between the mother and the fetus. D. Many species of bacteria do not cause disease. Bacteria are very important in many biological processes. State the names of 3 natural processes involving bacteria that are important to the ecosystems. Of course, these are the decomposition, fermentation, and nitrification. 5. Fires release carbon dioxide into the atmosphere. State one other natural process that releases carbon dioxide into the atmosphere. You can say respiration, decomposition, or you can even say volcanic eruptions. Double I, carbon dioxide is a greenhouse gas. State the name of one other greenhouse gas. Of course, this is methane. B. Data scientists use satellite images to analyze the occurrence of fires globally during a 14-year period. They tracked all fires that were larger than 0.21 kilometers square and therefore visible from space. Table 5.1 summarizes some of their data, categorizing the fires by location. The locations include natural ecosystems and land that is managed by people. The expansion rate is the speed at which each fire becomes larger. So here's the table with all the data. Using all the information in table 5.1, compare the data for the 2 managed locations with the data for the 3 natural locations. Okay, so over here we can see that the total number of fires in the natural locations over here, they are much more than the managed land. So we can say the first point is that the total number of fires in the natural ecosystems is higher than the total number of fires in managed land. We can also see that the natural savanna, it takes up a lot of number of fires over here. It has a much larger total number of fires, more than both of the managed land, like both of them. It has a total that is higher than both of these managed land. So we can also mention that point. You can also say that the fires that are being managed, they have a smaller average expansion rate. Over here we can see that in the natural fires, we can see that expand at 0.6, 0.4, 0.7, but the managed land has lower expansion rates. It's 0.3, and they also have a lower duration of fires. Over here they are 3.8 days, 3.4 compared to 4.6 and 4.1. Of course, this is lower than the natural, so we said till now 4 points. You can also say that there is more fires in the deforested areas more than the natural areas. So over here we can see that alone in the deforested areas there is over 900,000, and in the natural ones there is lower than 200,000 in each forest. So these are points you can write down. Double I, describe how the data in table 5.1 could be used to estimate the total area that was burned during the 14-year period for each location. So in double I, we're going to be multiplying the total number of fires, and we're going to also be multiplying with it the average expansion rate, and also you're going to be multiplying the average duration of fires. So the three of them together, you will get the total area that was burned during the 14-year period. Triple I, burning large areas of a forest is the cause of habitat destruction. Describe the possible consequences of destruction. So, of course, this means if there is habitat destruction, this means that there will be extinction of some um, animals. So, for example, if let's say a monkey lives on a tree and that tree is burned down, then the monkey will not be able to live on trees if there is no other trees, so they will go extinct. Also, there will be some plants and some like other organisms, they will go extinct as well. There will be loss of biodiversity, and there will be loss of genetic diversity as a result of the loss of biodiversity. And also there will be a negative effect on the food chains or the food webs, because for example, if we have a food web that depends on, for example, a specific type of plant and that went extinct, then the food webs will not be able to continue on. And there will also be a reduction in the population sizes of organisms. There is a lot of points you can write down in this question. 6a, figure 6.1 shows part of the human gas exchange system, and it shows us this image. Table 6.1 shows the names of some parts of the human gas exchange systems, their function, and the letters in figure 6.1 that identifies the parts. Complete table 6.1. So the first thing that we have here is A, which is the hair in the nose. This actually acts as a barrier to, and it also has mucus. This acts as a barrier to prevent dust and bacteria from entering and preventing some infections when you are breathing. So this traps particles and it acts as a mechanical barrier. Right here we have the prevents collapse of the airway. Of course, we all know that this is the cartilage. You can say that it's J or B. So this is either this one or that one. Contracts to decrease the pressure in the thorax. This is, of course, the external intercostal muscle, and we all know that it is located right here. And the diaphragm, it does the opposite, and it contracts to reduce the pressure and increase the volume in the thorax. So this is actually C. Protects the lungs from mechanical damage. This is the rib cage, of course. We're going to be, this is the rib cage over here, and this is the diaphragm, C, right here. Contains cilia to move mucus out of the airway. This is the trachea or bronchi or the bronchiole. J, B, or G. We have used J or B before, and this is okay because you can use it more than once, a letter. So if you used it before, it's okay to use it again if it's correct. Site of gas exchange. This is the alveoli, which is found at H. Double I, describe and explain how the alveoli are adapted for gas exchange. Number one, we can say that it has thin and one cell thick, because as we know that diffusion is fastest when there is a lower distance. So the one cell thick or the thin walls will allow for a lower diffusion distance. There is a large surface area which reduces the diffusion distance as well, and good blood supply to provide oxygen and remove the waste gases. BI, explain the differences in composition between inspired and expired air. In the inspired air, as we all know, that it has more oxygen, much more oxygen than carbon dioxide entering. The oxygen, it is required for respiration, and in the expired air, it has much more carbon dioxide and it has less oxygen. Double I, physical activity changes the concentration of carbon dioxide in the body. State where this change is detected and how the body responds to this change. If we do a vigorous activity, there will be respiration, and this respiration will cause carbon dioxide change in the blood, which means there will be more carbon dioxide. And this is detected by also the brain. So the response is increasing the depth of breathing and increases the heart rate. These responses will allow more oxygen to flow, and it will also, the increased heart rate will allow more blood to flow to take out the carbon dioxide and provide more oxygen. Triple I state the name of a solution that can be used to test for the presence of carbon dioxide gas. It is lime water, or you can say the hydrogen carbonate. So this was the end of our exam. Thank you guys for watching. Make sure to subscribe, like, and comment if you have any questions. And see you all in the next video.